Then in meiosis two meiosis goes around twice. That's how you wind up with haploid gametes. Okay? So you started with that parent back here, and in meiosis one, the two ranks of bow ties split apart. That's meiosis one. And it's in meiosis one that gene shuffling can occur. Okay? In meiosis two, the halves of the bow ties get pulled apart. And so finally you wind up with haploid gametes. Okay? And so you can see why one of these can get together with another one of these, and you would wind up with diploid again. Okay? And you can see the huge number of variations of individuals that you can get out of that with these gametes produced by meiosis. Okay? So here, we can apply our 2 to the n minus 1 rule. We have 3 chromosome pairs. So 2, the n is 3, and 3 minus 1 is 2, 2 to the 2 is 4. So suppose that mom is the pink one and dad is whatever color that is. You can see that there's 4 different ways that you can arrange those. So 4 different individuals that you could get in meiosis, in your metaphase lineup in meiosis 1. So meiosis is a very special form of cell division where you have this gene shuffling going on, and it goes around twice, meiosis 1 and meiosis 2, and you start with 1 cell and you wind up with 4 haploid gametes. Okay, so we start out with what's called a spermatogonium, which is diploid. 2N means diploid. Meiosis 1, and then you go through meiosis 2, and finally you wind up with the sperm. And the cytoplasm is stripped away, and there you go. We had 1 original cell giving rise to 4 haploid sperm. Same thing happens with eggs. Only with eggs, we don't want 1 oogonium doesn't give rise to 4 eggs because there isn't room for them all. Because a woman is born with all the oogonia that she's going to have. And so as you go through meiosis 1, one of the packages of chromosomes is discarded, and as you go through meiosis 2, all the other packages of chromosomes are discarded except one you keep as your haploid cell, because you don't want to expand the ovary 4 times its size, you know. So 1 oogonium gives rise to 1 egg plus 3 what are called polar bodies, little bags of genetic material that are discarded. So anyway, okay. So these pictures of meiosis that are shown in textbooks are confusing because they only show it with 2 pairs, and plus they're kind of hard to follow. Anyway, so if you don't like that, don't bother with it. My pictures, that's why I took the trouble to draw those pictures out. So diploid means having pairs of chromosomes. Haploid means only having 1 of each type of chromosome. Meiosis gives rise to haploid gametes. Human gametes possess 23 single chromosomes, 1 of each kind. And when 2 gametes combine, a diploid zygote results. Okay, so let's. The rest of the quiz next week is going to be genetics problems. Okay, and so let's start by looking at genetics problems that involve dominant and recessive genes. Okay, the first type of problem we will address will be one for which you have a dominant allele, and so it is the one that is expressed, and the other one, it's like you don't even know it's there. Okay? And so in heterozygous situations, the dominant allele is always expressed. The dominant allele is always uppercase, shown as an uppercase. So, okay, by deduction, do you think that the allele for Brown is lowercase or uppercase?
Lowercase.
Lowercase, and it's recessive, isn't it? So you see, these are the various genotypes that if you had two heterozygous parents, then this isn't very clear, but this is what the distribution you'd wind up with. This is homozygous recessive. Okay, so, no pens on these quiz, red or otherwise, no pens. Because if you are a superhuman if you don't have to erase one of these genetics problems. Get a pencil that has not some worn out little old eraser that doesn't work, but has a brand new eraser that works really well. Okay, if you don't have one like that, make a trip to Freddy's between now and the next quiz and pick up a couple and have them nice and sharp and all that. Because if there's a bunch of, if you use a pen and if there's a bunch of scribbling out and everything, it's bound to be reflected in your grade. If I can't understand it, how can I grade it? So use a pencil so that the final product is what you intend it to be. So, here's a real life problem. And I don't know if you've ever known anyone with cystic fibrosis. I have known a person. And luckily, the gene for cystic fibrosis is recessive. And a normal woman, heterozygous for this trait, you've heard the term carrier. If you're heterozygous in this trait, you are a carrier of that trait. So two heterozygous parents are going to, and this is the common scenario, two heterozygous parents marry. So what is the probability that their child will be normal, no cystic fibrosis?
75%?
Well, yeah, because you remember the result of those other problems. So anyway, here is one parent, the mother, and she has this genotype. And here's the father who has that genotype. And so there's a 50% possibility the sperm will look like that and a 50% like that. The same thing with the eggs. And so what you do is set up what's called a Punnett square, which are just little probability predictors. Okay? So of the female, there's a 50% chance of this allele being in the egg and a 50% of that. And in the sperm, 50-50. So then you just do the crosses. So that's that cross. And here we have this cross. Here we have that cross. And over here we have that cross. Are any of these individuals going to have cystic fibrosis?
Yes.
Yeah, the homozygous recessive. So 1 out of 4 boxes, there's a 25% chance that that their child will have cystic fibrosis. Was that the question? No, the question was, what is the chance it'll be healthy? 75%. But since they're not having a statistical cross-section of children, they could have 10 in a row. And they could all be healthy or they could all have cystic fibrosis. And every time you have a new kid, the odds start all over again. However, if you ask the question in advance, if you may recall that the second question there is what is the chance that their first 2 children in a row? If you ask that question before they've had the 2 kids, in statistics, which is what this is kind of a form of game theory. And you just use these little boxes to calculate the percents. And it's all in how you ask the question. in statistics. So if it's the first 2 children in a row before you ever had even one, then you multiply the probabilities times each other. What are the chances that their first 2 children in a row are gonna have cystic fibrosis? It's 0.25 times 0.25. It's a quarter times a quarter. What is a quarter times a quarter? 1/16. Guys aren't very good in your fractions. So there's a 1/16 chance that 2 children in a row are gonna have cystic fibrosis. However, if it's just their next child, after you had the first child, it doesn't matter whether he had cystic fibrosis or not. You start all over again with the next one. But if you're asking 3 in a row, before you ever had the first one, anytime you can say in a row, then you multiply them together. So it's all in how you ask the question. I'll always be careful to say in a row. Okay? So this is how you do these Punnett squares. And the first thing you do, you might as well, when you see one of these problems, is just draw the blasted square. Okay? So we saw that it was 75% chance of having a healthy child, 25% cystic fibrosis. But if it's 2 in a row, then it's 0.25 times 0.25. So, here's another one. In humans, the gene for tongue rolling ability is inherited as a dominant trait. You know what rolling your tongue is?
Mm-hmm.
Can you roll your tongue?
No.
You can't? Oh. Most people can, but because it's a dominant trait, see? But you must be homozygous. You can't do it?
No, she
Can't
can
do
do
it?
it. She can do it 3.
She can what?
She can do it 3.
Really?
Yeah.
Oh, yeah.
That's impressive.
Wow.
3?
Well, I'm not gonna touch that one with a 10-foot pole. Okay.
Yeah.
Wow.
So this is different. Usually the trait we're interested in is recessive, but this one is dominant. So if a woman able to roll her tongue, and we have to be told that she's heterozygous because it's the dominant, you don't know if you're not told, mates with a man who is not a tongue roller, do we have to be told if he's homozygous or heterozygous?
No.
No, because
He's
his phenotype is the recessive phenotype. And so we know he's little r. And so we know what the 2 genotypes of the parents are then. And so we could do the Punnett square. So here's one parent and there's the other. Okay? Homozygous dominant, homozygous recessive. So that's how we do it. Homozygous dominant, homozygous recessive. And so what was the question? I forget.
If the woman is able to roll her tongue.
What is the probability their child will be a tongue roller?
50%.
It is 50%. 2 out of 4 boxes. What is the probability their first 3 children will be tongue rollers? So if it's 50%, 0.5 times a half times a half times a half is an 1/8. Okay? So there's an 1/8, it's 12.5%, but I will take an 1/8. Whichever. Okay? And so 12.5%. The genotype for a non-roller, so the probability of a non-tongue roller is 50%. Okay? So that was problems where you had dominant and recessive situations. One, if you have a dominant gene present, it's just as though the other one isn't even there. Okay? What if both genes make a contribution, both alleles make a contribution to the phenotype? This happens very often in the case of pigments. Many pigments in nature are made out of proteins, and so they're coded for by genes. Okay? And so, pigments are kind of interesting. We can call this, some people call this codominance, incomplete dominance, whatever. The best thing to do is just remember this type of situation, which one, you know what I'm saying? Just remember the different problems of which ones were dominant recessive and which ones were incomplete dominance. So, there's a gene for straight hair and a gene for curly hair. If you have one of each, you'll have wavy hair. Doesn't that make sense? Kind of halfway in between curly and straight. Here it'll be kind of kinky. Here it'll be totally straight as an arrow, and here it'll be wavy. So those are the various genotypes for the various phenotypes. The phenotype and the genotype. So we'll maybe work a problem with that later. Certainly with blood antigen genetics, each one makes a contribution, kinda. I mean, if you got the gene for if you got the A gene, your cells will have the A antigen. If you have the B gene, you have the B antigen. So if you have both of them, then your cells will have both antigens. Okay? So in that regard, you could say they're codominant. So type A, you're either heterozygous or homozygous, and you'll still be type A, either one of those. Type B, with type B, AB, you can only have one genotype. And type O is just the absence of either one, the gene for either antigen. You have no antigens on your blood cells. Okay? And there are more than 50 different blood antigens. This lab, for some reason, mentions the M and the N blood group. You treat them just like the A and the B. It's easy. Okay. So they're the three different genotypes. And you handle them just the same way. The Rh factor, okay, and that's of interest. Rh positive, you can have one of two different genotypes. See, we never mentioned that back last quarter, did we? So, and the outcomes that you can get will be different. If the father is this one, then there's about 100% chance, as I recall, there's a huge chance that the fetus will be Rh positive. Whereas if the father is this one, there's not 100% chance. And of course, Rh negative is basically having the absence of the gene for that Rh antigen. Okay, so if one parent belongs to blood group A and the other to blood group B, is it possible for their child to belong to any of the four blood groups? What's wrong with this problem?
Well, because it's gotta be one of them.
Well, what's lacking in this problem? Let me ask it that way.
Well, it doesn't say recessive.
We're not being told whether the parent is AA or AO. And the same here. So you've got to do all the possibilities. Anytime that you're not given that information, and that'll crop up on quizzes in here. Okay, so type A could either be that or that, and type B could either be that or that. So you got to cross that with that, that with that, that with that, and that with that. So you got to do 4 Punnett squares. Here we do the one with all homozygous, and you get all ABs. What was even the question? Can any of the offspring be type O? Was that it?
No, they're gonna be any of the
Can
four.
they be any of the four?
Okay, yeah, okay, so we certainly get type AB out of that one. So here we're getting type A, B, and type A. We're doing a homozygous and a heterozygous, okay? We're doing the mom homozygous and the dad heterozygous. Here we do the mom homozygous and the dad heterozygous. And we get type A, B, and type B. Finally, we get a type O out of this one, where they're both heterozygous. So you can indeed get all four types, can't you? So all four types are possible. Okay, both parents must be heterozygous. You might have guessed that from the onset. The allele for curly hair is codominant to the allele for straight hair. If a woman with wavy hair mates with a man with curly hair, what is the probability their child will have straight hair? Okay, well, here's the wavy hair and here's the curly hair. Okay, so here's wavy, curly, and what was the question?
Will they have straight?
Will the
What is the probability that the child will have straight hair?
straight hair, is there any, will you
No.
have any straight hair? No. No. No straight hair there. Straight hair would be SS. Okay? The genotype for wavy, so the probability of wavy hair is 50%. Straight hair is not possible. Okay? So, you kind of got to be a bit of a detective sometimes to try and figure out the genotypes of the parents. But all the information should be given. And as I'm trying to say with this last slide, sometimes it's more, we're making it simpler than it really is. So with that said, let me go over to the board and work some of these problems. Okay? And the ones I'm going to work are off these practice problems. Did anyone print out the practice problems? No. Okay, could you get the lights for me? Lab, but the ones I'm gonna work are on here. Because these are kind of interesting, a couple of them. Okay? Okie dokie. Especially on the second page here. The chicken problem. So consider a situation wherein there are chickens of 3 different colors. Okay? The genotype for black is BB, the one for blue is big B, little b, and the one for white is little b. What it really is, in these pigment problems, is that there's a functional black pigment that is denoted by uppercase B. That's a fully functional, the gene works for the black pigment. And if you have 2 of those genes, you're gonna have a super black chicken. Okay? If you have only 1 functioning gene, as in the case of, as in this case, then you'll only have 50% as much black pigment, won't you? And so the chicken will be kind of a light black, which could be construed to be blue. Get it? And if you have this genotype, see these pigment genes get mutated. You have no functional gene for black pigment, so the chicken appears white. And so, what is the probability of obtaining a white chicken if a black chicken is crossed with a blue chicken? So, what you do, these are kind of fun. So, a white, if a black is crossed with a blue, and we want to know if you can get a white, my guess would be not.
Yeah.
So we have, so that one's gonna be B, 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 B. So 50% black and 50% blue, okay, but no whites. Get it?
Mm-hmm.
No? Got it? Okay, what is the probability of obtaining a blue chicken if a black is crossed with a white? My guess is you could do it, but we'll see. So we're crossing a black, so you see why you use a pencil? Okay,
Okay.
we're crossing, it's of a blue chicken, a black with a white, black with white. So we're gonna get B, B, oh, B, B, I was wrong. That's why I always gotta do the square. I wouldn't bet a nickel on any of these before you do the square, cause I guessed that you could do it. What was the question?
If you
Of
can get a
oh.
blue chicken,
You
you
can
can.
get
Oh, yes, you can, you
100%.
get all blue chickens. Well, I thought it was white chicken, okay.
That was the.
Gotta read the problem now, see. Of course, codominance is operative in this problem, and there's all kinds of other variations that you could ask there. So the chicken problem is kind of fun. Assume that there are 3 colors of rabbits. That should be an S at the end, rabbita. Rabbita is plural for rabbit, see? And colors is misspelled. I think I was in a hurry when I zapped this out. Collars. So we have, and so you can have rabbits that have kind of a mottled pattern on them. I use this for the chinchilla one. 3 colors of rabbits, full colored brown, chinchilla and white. Further assume that the full colored allele is dominant. So the full colored would be this one. It is dominant to both the others, and you gotta be careful. I use a little teensy c for white, okay? So this one is dominant to both of the others, and this one is dominant to this one. So we've not run into anything like that, and it's the only case where we will. But it makes for some interesting problems, okay? So if a white rabbit is crossed with a full colored rabbit, it is found that 50% of the offspring are full colored and 50% are chinchilla. What are the genotypes of the parents? So you see, we know what the genotype of a white rabbit is crossed with a full colored. We know that the white one had to be little c, right? But we don't know, since this one dominates over that one, we don't know whether this full colored individual was homozygous or heterozygous. This one could have been big C with this or with that. And in both cases, you'd get a full colored rabbit, wouldn't it? Okay. So they told us though that 50% of the offspring were chinchilla. Okay. And so 50% of the offspring were chinchilla. Okay. They had to have come from that, and this had to be, this thing is dying on me. Okay, so the genotype of that parent was the fact that you got 2 chinchilla offspring told me that the other allele had to be that one for this parent, because this dominates over both of them. So this one could have been the same as that, it could have been this one, or it could have been a little c, and you still get a full colored rabbit. But by telling us that 50% of the offspring were chinchilla, they told us that chinchilla had to be in here somewhere. Okay? So these are genotypes of the parents. One parent is that and the other parent is that. Get the gist of that? Okay. So, what's the next one? If a white rabbit is bred to a heterozygous chinchilla, what is the probability that the offspring will be chinchilla? Okay, and so we have a white rabbit. So we know it's heterozygous chinchilla. What's the other allele gonna be though, heterozygous?
A little C.
It's gotta be a little C, cause if it was a big C, then this wouldn't be a chinchilla anymore. Cause big C dominates over that. So we know what the genotypes of the two parents is, so we know that we're gonna get we're gonna get 50% white and 50% chinchilla. So what was the question? What will be the, okay, 50% chance that they'll get a chinchilla offspring, because chinchilla dominates over white. Okay? There's all kinds of other chinchilla problems. I can't, you know, you can just think up your own variations. Okay, and there'll probably be one on the quiz. So those are the chinchilla ones. What about Hereford cattle? Okay. The gene for horns is actually recessive to the hornless one. So two heterozygous polled Herefords are crossed. So little h has got to be horns. Two heterozygous polled Herefords. So if they're polled, you have to be told that they're heterozygous, because otherwise you wouldn't know, would you? So they're heterozygous polled, are crossed. Two heterozygous polled are crossed. So they're both heterozygous. Well, anytime you cross, oops, I did that wrong. Blasted. Sorry about that. That's why you have an eraser.
Not anymore.
Naughty, naughty. Okay. Anytime you have two heterozygous parents, there's always a 25% chance of homozygous.